myotonic dystrophy is caused by a repeat expansion at the end of one of those genes. And that repeat expansion sequesters RNA binding proteins and prevents cells from splicing proteins the right way. So the way I explain that to my patients is that your cells, when they start making proteins, they it's like a clothing factory, and they have squares of fabric, and you have pairs of scissors that cuts the squares of fabric into shirts or pants or dresses. And your cells do the same things. They have to cut proteins to the right shape. In myotonic dystrophy, that pair of scissors is bound up by that repeat, and so the cells don't make the right shape proteins. They just have squares of fabric. And so we know from animal work and cell work that if you get rid of those repeats, you can release that pair of scissors to do its job and allow the body to function potentially normally. And so the Avidity Biosciences has developed an siRNA molecule that's designed to bind and knock down that DMPK repeat. So I'm excited to see kind of whether or not it had any effect in that in terms of RNA splicing and function. The primary objective of this first study, though, was safety and tolerability. It's the first time this particular drug was tested in humans, and so the main goal was to assess the safety and tolerability of it. Yeah, so this trial was a small study with about 8 to 12 patients in each treatment group plus a placebo, which was combined across the group. And so, as I said, I think the results were really better than expected. But now it needs to move into a confirmatory or phase 3 clinical trial to show those benefits across a larger patient population. And so I'm aware and believe the company is moving forward with that phase 3 clinical trial and are discussing how best to design, prepare, and execute it with appropriate regulatory authorities.